welcome, respected Director Sir. I, Prabha Rajput, welcome you all in the today's expert lecture series. Today we have Dr. Ashok Jangra Sir in our expert series as an expert. Director Sir, please welcome to the Dr. Jangra Sir.
So thank you very much, Dr. Pirva. On behalf of ISF Dialogue Series and ISF College of Pharmacy, I welcome Dr. Ashok Jangra ji from core of my heart, and I also welcome whole team of the ISF Dialogue Series. So first of all, I just give the brief about this, the purpose of Dialogue Series and what is the need. So ISF College of Pharmacy decided to invite some of the experts, those who are have the quality of the research and education on this platform, and we can discuss some of the important topic, and it will be the helpful to our youngster associates and some teachers as well as the research. So the purpose behind of this one, we have number of the lecture available on the YouTube channel, but the purpose behind of this one is the special, some of the specific lecture, invited lecture we can put on the our YouTube channel, as well as we can also call some of the, you can say, queries why the students, as you know that pharmacy profession is growing very fast, but the availability of the experts, teacher and researcher, they are not sufficient in the education system as well as the research, so that we want to provide the facility common platform to each and everyone. So this is our basic aspect. So I again welcome from core of my heart Dr. Ashok ji, and thankful to our organizing committee member for this interaction and involved to me in this ISF dialogue series. So over to Dr. Birbha.
Thank you, Director Sir. Thank you so much for your kind words. Again welcome Dr. Ashok Jangra Sir. He has he has done his master and PhD from Naipur Mohali and Naipur Guwahati. He has also awarded lots of grant for the foreign and the India as well. And now he is working as assistant professor in the Central University of Haryana. He is an expert in the neurodegenerative disease. So I warmly welcome Dr. Ashok sir to proceed his lecture. Dr. Ashok sir, please proceed.
Thank you, Dr. Prabha. Am I audible?
Yes, sir.
Okay. So, first of all, I would like to thank Professor G.D. Gupta sir for inviting me to deliver a lecture in this lecture series. So, let's begin the lecture. My slides are in presentable mode or not,
Yes, sir, it's
Dr.
presentable.
Prabha? Okay, okay. So, in this lecture, we are going to talk about the interplay between the autophagy, oxidative stress, and the ER stress, that is endoplasmic reticulum stress, in neurodegenerative disorders. So let's start with the autophagy process. So as you know, autophagy means self-eating, means the autophagy process involves its own macromolecules, its own organelles of the cell. So the main objective of this autophagy process is to break down the cytoplasmic components like macromolecules and the cellular organelles. And why this autophagy is important? Because it is useful for the cellular metabolism as well as to ensure the cellular homeostasis. Okay, then as you can see in this picture, figure number 1, the autophagy can be divided into three types: macroautophagy, microautophagy, and chaperone assisted autophagy. So first of all, we are going to discuss macroautophagy. So macroautophagy is a highly conserved cellular process that involves the formation of autophagosome. As you can see in this figure, the pre-autophagosome interact with the cellular macromolecules, which are going to be digested by the lysosomal enzymes. So the interaction between this PAS and the cellular macromolecules form the autophagosome. And This autophagosome subsequently interact with the lysosome and form the autolysosome. And this autolysosome is the actual site where the different organelles or different cellular molecules are degraded. So this is all about the macroautophagy. On the another hand, the microautophagy is completely different from the macroautophagy. So in the microautophagy, here there is simply engulfment of the vesicles by the lysosome by a process called as cytoplasmic invagination. And the third type that is very selective, it is called as chaperone mediated autophagy, CMA, or it is called as chaperone mediated autophagy. So it is a highly selective autophagy in which only specific substrates are digested. So what kind of specific substrate? Those substrate which have C terminal that contains KFERQ motif in their structure. So these type of motifs that are present with the macromolecules are easily recognized by the chaperone proteins that are heat shock proteins. So these heat shock protein interacted with the substrate molecules, and then the complex of the heat shock protein and the substrate presented to the LAMP2A protein, and this LAMP2A protein is present on the lysosomal surface. So LAMP2A resulted into the degradation of the cellular substrate or the molecules or any type of organelle, right? So now coming to the topic, how these autophagy process is involved in the neurodegeneration, whether this autophagy increases the neurodegeneration or autophagy uh, reduces the neurodegeneration process. So in the table number 1, you can see that there are two type of autophagy, basically constitutive autophagy and inducible autophagy. So constitutive autophagy, as you can see, this constitutive autophagy, the main objective of this autophagy is to maintain the cellular homeostasis. Means this type of autophagy replaces the old proteins and organelles with new ones. Okay. However, in case of inducible autophagy, as the name suggests, inducible means this type of autophagy is induced by certain stimuli like food, like nutrient deprivation, or whether there is this exposure of certain toxic chemicals. Then this type of autophagy can be induced, and the objective of this inducible autophagy is to this cellular adaptation to the condition like nutrient deprivation. So this inducible autophagy can also damage the DNA. So now coming to the relationship between the neurodegenerative disease and the autophagy. So normally, when the newly protein is synthesized in the endoplasmic reticulum, then they are folded in the endoplasmic reticulum. But some of the proteins are misfolded, and this misfolded proteins form the inclusion bodies. So normally, under the homeostasis condition, in the absence of any neurodegenerative disease, this misfolded proteins and the inclusion bodies are easily degraded by the lysosomes by the autophagy process. However, if in case the autophagy is suppressed by several reasons, then there is a increase in the level of these inclusion bodies, specifically in the neurons, and this inclusion body deposition in the neurons causes neuronal degeneration. Now the neuronal degeneration can be takes place. So this autophagy can be regulated by several mechanisms. We will discuss in the later slide how this autophagy can be regulated by the all the three arms of the endoplasmic reticulum stress mechanism, or how the oxidative stress is involved in the regulation of autophagy. So there are numerous signaling pathways that are involved in controlling the autophagy. So among them, the mTOR complex, that is very important. So this mTOR complex is the main regulator that integrates the inputs from the growth factors, hypoxia, ATP, and insulin signaling. So mainly, whenever the mTOR is activated, then there is a inhibition of the autophagy. So specifically, this the when we talk about this types of autophagy, macro, micro, and chaperone mediated, the macro autophagy is the most prominent one, and this macro autophagy starts with the activation of ULK complex. So ULK complex formation is essential for the initiation of the autophagy process. So it is a very crucial process.
Sir, I think the network at his side is lost.
Network problem.
Yes.
So, Madam Prabhaji, you can take phone call and we
Yes,
can
sir. He will
arrange
join
for.
within 2 minutes. He messaged me.
Okay, okay, okay.
Yes, sir.
Okay, Dr. Prabha. So we are talking about, yeah, am I audible?
Yes, sir.
Okay. So we are talking about how this autophagy is involved in the neurodegeneration process. Okay. So some researchers found that the autophagy failure is the uh, primary underlying cause for the neurodegenerative diseases. So, for example, in the most neurodegenerative diseases, including amyloid lateral sclerosis, Parkinson disease, Alzheimer disease, These accumulation of these misfolded protein is a common, you can say, pathological hallmark for the neurodegeneration. So these misfolded proteins damage the neurons and eventually causes neuronal cell death. So because the reduction of autophagy activity directly affects the accumulation of misfolded proteins, thus autophagy can be considered as a key target for the treatment of the neurodegenerative diseases. So now moving to the oxidative stress. So oxidative stress is a very well-known term because oxidative stress is involved in almost in every disease or in every pathological condition. So as you can see in figure number 3, the ROS are generated by enzymatic as well as non-enzymatic reactions. So these ROS reactive oxygen species are produced under normal condition as well as under the pathological condition. So whenever these reactive oxygen species are produced like superoxides, O2 negative. So this superoxide anion is a very dangerous reactive oxygen species that can negatively affect the proteins and various other biomolecules. So in our body, in our cell, superoxide dismutase is present that converts superoxide into the hydrogen peroxide. But eventually, due to the Fenton reaction, the hydroxyl ions or oxygen species, hydroxyl species can also be produced that can also damage the biomolecules. So it means the superoxide as well as the hydroxyl radical ions can also damage the our lipids, our proteins, or our DNA. Or high amount of oxidative stress or prominent oxidative stress can also impair the mitochondrial function. Moreover, under the oxidative stress condition, the nitric oxide synthase activity is also increased. So whenever the nitric oxide synthase activity is increased, there is a production of nitric oxide in the cell. So the nitric oxide again is a reactive nitrogen species. So now, when inside the cell, the level of oxygen, level of the peroxide this superoxide as well as the nitric oxide is increased, then they react with each other and form the peroxynitride. As you can see here, this is peroxynitride species, and this peroxynitride species is more powerful reactive oxygen or nitrogen species as compared to the nitric oxide alone or the superoxide alone. So these readily damage the our various proteins or the lipids. Moreover, our body has endogenous antioxidants like glutathione. So glutathione readily converts the superoxides into the non-toxic or non-reactive oxygen species. So so the question is how these reactive oxygen species are generated under the normal condition or under the pathological condition. So inside the mitochondria, electron transport chain is continuously activated. So here there are different complexes in the mitochondria, complex 1, 2, 3, and 4. So specifically in the neurodegenerative diseases, the complex 1 is dysregulated. The researchers found that the complex 1 is dysregulated in the neurodegenerative disease, and this complex 1 is specifically responsible for the reactive oxygen species generation. Now coming to the role of oxidative stress in the neurodegeneration. So as you can see in this figure, the oxidative stress is a condition where there is an imbalance between the antioxidant and the reactive oxygen species or the reactive nitrogen species. So whenever there is an increase in the reactive oxygen species in comparison to the endogenous antioxidant, then this condition is called as oxidative stress. So whenever the oxidative stress occur in our body, then then the biomolecules like proteins, lipids, DNA, RNA, certain enzymes are very sensitive to these free radical ions. So the free radical ions causes mitochondrial dysfunction, or the free radical ion activate the astrocytes or the microglia inside the brain, and the reactive microglia, the microglial cells, releases the inflammatory cytokines, and these inflammatory cytokines eventually damage the blood brain barrier, as well as they also produce the neuronal apoptosis. So it means the oxidative stress generated by the reactive oxygen species can eventually causes neuronal apoptosis, can eventually causes cell death, production of Lewy body, deposition of Lewy body, or deposition of amyloid plaques inside the cell, or there is a increase in the phosphorylation of tau. So it means the misfolded proteins are easily accumulated inside the neurons, inside the brain, and as well as the apoptosis, the neuronal cell death is also increased under the influence of oxidative stress and oxidative stress induced inflammation. Right? So now coming to the most important pathway, that is endoplasmic reticulum stress. So endoplasmic reticulum stress, as you can see in figure number 5, when there is a rise in the level of unfolded protein. So as you already, I think you already know that endoplasmic reticulum is a site for the protein synthesis, as well as the protein folding. So whenever the newly synthesized proteins, they are folded inside the endoplasmic reticulum. But under certain conditions, either the protein folding capacity of the endoplasmic reticulum is decreased, or there may be chances that there is an increase in the load of the protein folding. So when there is an imbalance between the protein folding capacity of the ER and the protein folding load of the ER, then this condition can cause ER stress. So ER stress may be protective or it may be destructive. So it depends upon the intensity of the stress. So in the ER stress mechanism, there are three arms. There are three proteins which regulate the ER stress mechanism. So these three proteins are PERK, ATF6, and IRE1. So under homeostasis means when there is no protein folding overload on the ER, then GRP78 regulates these three types of protein. But under the condition when there is a increase in the amount of unfolded protein inside the endoplasmic reticulum, then the GRP78, or we can say the other name of GRP78 is BIP. So this GRP78 or this BIP detaches from these three types of proteins, and this detachment lead to activation of all the three arms of the ER stress. Means after detachment of GRP78 under the influence of unfolded protein accumulation, the PERK, the ATF6, and the IRE1 are activated. And these three arms activation causes certain protective responses, or they produce certain destructive responses like apoptotic cell death via CHOP. So let's understand how this PERK is involved in the apoptosis, or how this ATF6 is involved in the reducing the protein folding load in the endoplasmic reticulum. So as you can see here, this PERK activation leads to the phosphorylation of eukaryotic initiation factor 2. So when this phosphorylation takes place, then there is first thing is there is reduction in the translation. It means there is reduction in the formation of proteins. Secondly, EIF2 alpha phosphorylation activate the ATF4. So this ATF4 activation regulates certain gene expression. So gene expression like CHOP expression or the genes which are responsible for the stress response or for the redox reactions. Next, the second arm of this ER is ATF6. So whenever this GRP78 detaches from the ATF6, so ATF6 cleaved inside the Golgi body and the cleaved form transported inside the nucleus. So here this ATF6 again regulated the chaperones which are involved in the protein folding. It also increased the expression of CHOP which is involved in the apoptotic cell death, and it also increased the mRNA level of XBP1, and this XBP1 mRNA is involved in the IRE1 signaling. Now, moving to the third arm, that is IRE1. So, IRE1 activation leads to the cleavage of XBP1 mRNA. So, there are certain modifications occur under the influence of IRE1 that leads to the formation of soluble form of XBP1 mRNA. So, this soluble form of XBP1 mRNA migrated inside the nucleus and regulate the certain gene expressions, like they increase the chaperones like P58, which is involved in the protein folding, and for the genes which are involved for the protein degradation. It means IRE1 reduces the protein folding load over the ER. So how? First of all, the chaperones are involved in reducing the protein folding, involved in the protein folding, and those proteins which are accumulated in the unfolded form are degraded by certain enzymes. So this type of degradation is called ERAD degradation, ERAD pathway of ER stress. So all these three are the arm of ER stress which are involved in this ER stress mechanism. So now coming to the mechanism of ER stress, how this ER stress is involved in the neurodegeneration, how this ER stress is involved in the pathogenesis of Alzheimer's disease or the Parkinson disease. So as we talked about earlier, that endoplasmic reticulum is involved in the production of survival factor or involved in the production of apoptotic factor. So whenever the endoplasmic reticulum stress is activated under the accumulation of unfolded protein, then there is induction of ER chaperone, there is induction of amino acid transporters, there is translation repression, or there is activation of ERAD component that we discussed a few seconds later previously in the previous slide, or the endoplasmic reticulum stress pathway can induce the CHOP enzyme, CHOP protein, and this CHOP protein is involved in the regulation or expression of death receptors that that we are going to discuss in the figure number 6b. So let's discuss how this CHOP is involved in the apoptotic cell death. So as you can see here, we are discussing only one arm of the ER, that is PERK, PERK. So under the influence of ER stress, PERK is activated, and this PERK activation leads to the phosphorylation of EIF2 alpha. And this EIF2 alpha eventually activate the ATF4, and this ATF4 further activate the ATF3, and these two, ATF4 and ATF3, both are involved in the activation of CHOP. So now the complex between the ATF3 and the CHOP regulate the expression of DR4 and DR5 that are called considered as death receptors. So this CHOP also activate the caspases, caspase 3, caspase 7. Moreover, they also activate the BEX and BAC. As you know, these are the apoptotic uh, caspases, BAC and BACs. So BAC and BACs increases the permeability of mitochondria. So what happened when the permeability of mitochondria is increased? There is release of the cytochrome C from the mitochondria, and this cytochrome C eventually activate the caspase 3, which ultimately involved in the apoptosis. Now coming to the uh, next slide that depicts the link between the autophagy and the oxidative stress. So in the previous slide, we discussed what is autophagy, what is oxidative stress, what is endoplasmic reticulum stress, and how these three processes are involved in the neurodegeneration process. So now we are talking about the link between the autophagy and oxidative stress, means how the autophagy influence the oxidative stress. So here you can see the ROS readily induce the autophagy mainly by mitochondrial hydrogen peroxide and the and the superoxide ions. So these ions may modulate the autophagy via mTOR dependent pathway. As you can see, the mTOR pathway, as we discussed earlier, this mTOR pathway is involved in the regulation of autophagy. So the oxidative stress, the reactive oxygen species regulate the autophagy process by mTOR pathway. So the ROS induced autophagy and the mitophagy both can abort the ROS for redox homeostasis. So whenever the autophagy is activated or initiated, then this autophagy as well as the mitophagy reduce the level of reactive oxygen species. So, in response to the high level of reactive oxygen species, the NRF2 keep keep1 and ARE response can also activate it. So, phosphorylation, as you can see, the phosphorylation of P62 by the autophagy can promote the integration of phosphorylated form of keep1 and ubiquitinated form of NRF2. Then this leads to the reactivation, or the NRF2 is translocated inside the nucleus, and then this NRF2, as you can see, this NRF2 leads interact with the ARE, and this ARE and NRF2 complex regulate the regulation of antioxidant genes. Now, this NRF2 activation leads to the activation of, or leads to the synthesis of those enzymes which are involved as a as an antioxidant, right? So by this pathway, we can say that the autophagy is considered as an endogenous antioxidant pathway, because after activation of autophagy, we can see that this autophagy as well as mitophagy both reduce the production of, or reduce the level of reactive oxygen species. So it means by inducing the autophagy, we can reduce the oxidative stress inside the cell, which can be beneficial in the treatment of neurodegenerative diseases. Now coming to the link between the autophagy and the endoplasmic reticulum stress. So how the autophagy and ER stress interplay with each other. So as you can see in the figure, all the three branches, PERK, ATF6, IRE1, all the three branches can directly activate the autophagy induction as well as the autophagy production formation during the ER through activation of certain ATG genes. So if you understand the detail of the macroautophagy, so you can see several type of ATGs are involved in the macroautophagy. So these ATGs can be induced by all these three arms of endoplasmic reticulum stress. So for example, the PERK mediated ATF4 translation induces the CHOP. It means this PERK can induce the apoptosis, as well as this CHOP can also increase the transcription level, increases the gene transcription of ATG genes, as well as those regulators which are involved in the autophagy. So the function of CHOP is not only to induce the apoptosis, but also to induce the autophagy by increasing the gene expression of ATG and the gene expression of certain regulators that are involved in the autophagy, right? Moreover, there are certain others like ATF6. As you can see, this ATF6 also regulate the autophagy by declin-1, as well as IRE1 also regulate the autophagy or induce the autophagy by baclin-1 and GNK. So it means there are multiple pathways, there are multiple molecules by which the three arms of the ER can induce the autophagy process in the cell. So it means by regulating the autophagy, by regulating the ER stress, we can regulate the autophagy, right? So now coming to the link between the oxidative stress and ER stress. So how these two are interlinked with each other in the neurodegenerative process. So during the ER stress condition, the misfolded proteins are accumulated in the endoplasmic reticulum, right? As we already discussed this point. So GSH, what will happen? The GSH, that is the glutathione, is involved in the reduction of the quantities of the misfolded proteins. So this process, this event can also decrease the amount of GSH in the cell. So on the other hand, certain enzymes like protein disulfide isomerase, flavin adenine dinucleotide, endoplasmic reticulum oxidoreductase 1 beta are also involved in regulating the redox balance. So, under the influence of oxidative stress, there is production of reactive oxygen species as well as the amount of glutathione is also decreased. So again, here what will happen whenever the oxidative stress occur, then there is a imbalance between the reactive oxygen species and the endogenous antioxidant. So this endoplasmic reticulum stress disturbs the redox balance and causes oxidative stress in the cell. Now coming to the crosstalk between these three processes. So earlier we discussed the interplay between the autophagy and oxidative stress, interplay between the oxidative stress and the endoplasmic reticulum, and the interplay between the autophagy and the endoplasmic reticulum. So now in this slide, in the figure number 10, as you can see here, the interplay between these three processes. So as you can see here, and whenever there is a oxidative stress, then what will happen? The calcium ions are easily leak out from the endoplasmic reticulum. So as I think you know already that the endoplasmic reticulum is considered as a reservoir site for the calcium. So calcium, excess amount of calcium is stored inside the endoplasmic reticulum. But under the oxidative stress, the calcium ions are easily leaked out from the endoplasmic reticulum and then they migrate into the mitochondria. And here, the high amount of calcium increases the reactive oxygen species. So, this reactive oxygen species further leads to the release of cytochrome C from the mitochondria as well as the APAF1, that is apoptotic response activating protein, activating factor. So this APAF1 as well as the released cytochrome C interact with the procaspase and convert them into the caspase 9. So now the activated form of the caspase 9 activate the caspase 3, and as you know caspase 3 is a executioner caspase, means this caspase 3 is ultimately involved in the neuron in the apoptotic cell death. So it means the reactive oxygen species and can induce the apoptosis. Now, the second one is endoplasmic reticulum stress. So, endoplasmic reticulum stress, all the three arms, ATF6, this PERK, this IRE1 as well as the PERK. All these three arms are involved in the apoptosis. The ATF6 arm, the spliced form, migrate to the nucleus. Then, in case of IRE1, here the XBP form, the soluble form of XBP1 migrate to the nucleus, inside the nucleus. And in case of PERK, this DDI3 or CHOP is migrated inside the nucleus to induce the apoptosis. It means all the three arms of the endoplasmic reticulum stress are involved in the apoptotic cell death. Moreover, these endoplasmic reticulum stress pathways can also regulate the autophagy, as we discussed earlier, by MAP kinase, JNK, as well as the ATF4, right? So all these three processes are interplay or interacted with each other and involved in the neurodegenerative diseases. Now coming to the next part, that is very important, that how these three processes are involved in the pathogenesis of Alzheimer disease. So what happen in the Alzheimer disease? Here, whenever there is rise in the reactive oxygen species, then reactive oxygen species increases the activity of certain kinases. Kinases like MAP kinase, JNK, GSK3, all these three type of kinases phosphorylate the certain proteins. So these kinases hyperphosphorylate certain proteins like neuronal proteins, tau protein, or the APP, amyloid precursor protein. So ROS hyperphosphorylated certain proteins, and due to the hyperphosphorylation of these proteins, these are easily accumulated inside the brain, and this misfolded so under mild condition, or under when there is very less amount of misfolded proteins. So these misfolded proteins can be easily degraded by the autophagy. But when there is a rise in the level of misfolded protein, or when there is a reduction or there is an inhibition of macroautophagy, then there is a chance of accumulation of misfolded protein inside the cell, inside the cells. Moreover, the reactive oxygen species can also dysfunctionalize the mitochondria, and that leads to the release of cytochrome C. So, as we discussed earlier, the cytochrome C as well as the APFA1 is also involved in the neuronal cell death. Moreover, the amyloid beta is a pathological hallmark for the Alzheimer disease. So, what happen in the Alzheimer disease when there is a amyloid beta aggregation? So, this amyloid beta aggregation inhibit the formation of autophagosome. So, it means amyloid beta itself accumulated in the brain, as well as certain other proteins are also accumulated in the brain in the absence of autophagosome formation. Moreover, the endoplasmic reticulum stress are also involved in the hyperphosphorylation of tau protein. So it means all these three, the autophagy is reduced in the Alzheimer disease, the reactive oxygen species hyperphosphorylated and misfolded the proteins, and the endoplasmic reticulum stress also hyperphosphorylated certain proteins and causes misfolding of the proteins. So all these three processes are involved in the pathogenesis of Alzheimer disease. Now coming to the interplay of autophagy and oxidative stress and ER stress in Parkinson disease. So as you can see here, the DDI3T, RIDD, and ATF6, which we discussed earlier. So these three are involved in the apoptosis. And how these are involved in the apoptosis? They are involved in the activation of caspase 3. Moreover, they are also involved in the production of chaperones. The production of chaperone leads to the reduction in the accumulation of unfolded proteins, because the main function of the chaperone is to fold the unfolded proteins. Moreover, in Parkinson disease, the alpha synuclein is easily accumulated. So, the major problem in the Parkinson disease is accumulation of alpha synuclein. So, this alpha synuclein is also interacted with the BIP, and it can also activate the all the three arms of the endoplasmic reticulum stress, and eventually also these three arms also eventually produce the apoptosis, and they also inhibit the formation of autophagosome. As you can see here, the reactive oxygen species can inhibit the autophagosome formation. So again here, in the absence of macroautophagy and in the presence of high level of reactive oxygen species and high level of endoplasmic reticulum stress, can produce the apoptosis in the Parkinson disease. Now, moving to the conclusion, the autophagy, oxidative stress, as well as the ER stress are considered as a double-edged sword for the neurodegenerative condition, because we discussed in the previous slides that sometimes ER stress act as a protective, or sometimes the endoplasmic reticulum stress induce the apoptosis, and sometimes the oxidative stress inhibit the autophagy, or the autophagy is involved in the reduction in the misfolded protein. So there is a strong correlation between the impaired protein homeostasis, autophagy dysfunction, protein aggregation, intracellular stress production, and the neurodegenerative disease. So a considerable number of therapeutic modulators have been developed earlier and evaluated under clinical trials, but they failed to produce the therapeutic response. So the main problem associated with these therapeutic modulators are low bioavailability, inability to cross the blood-brain barrier, and certain obstacles like pharmacokinetic parameters by which they cannot be efficiently translated into the commercial products. So that's all from my side. Thank you.
Thank you, Ashok sir. It is a very informative and knowledgeable lecture. It will help to all the neuroscientists who is working on the area of neurodegenerative diseases like Alzheimer's and Parkinson's. We are very thankful to you, sir. Thank you so much.
Thank you, Dr. Prabha. Thank you, sir.
Sir,
Thank
आप
you for
mute.
inviting me.
Sir, you are mute. GD sir.
So I